Welcome to Instant Biology by Dr. Neela. The topic that we would be dealing today is Sanger sequencing. This is a very important topic and is asked in multiple life science examinations. So, in Sanger sequencing, we would be dealing with what is the need of Sanger sequencing, what is the principle of Sanger sequencing, and also how do we read a sequence with the help of a Sanger sequencing, right? So let us go point by point and let us consider first of all that what is the need of Sanger sequencing, or let us address it in this way, that what is the need of sequencing primarily. So when you do sequencing of a DNA strand, you get a particular position of each nucleotide that is present in the strand. So what is the use of getting the single position of each nucleotide? How will you use it in your experiments? See, let us take an example and understand this. If a person is suffering from a disease, okay, and you want to understand, you want to know that which particular nucleotide or what mutation has caused this disease. In order to understand this, in order to address this problem, you will have to sequence that particular gene of the diseased person and compare it with the gene of a normal person, and then you would be able to know, you would be able to understand that which portion of a particular gene is causing this kind of a distress to the individual, right? So in order to understand the mutations, in order to know about the changes that have taken place in the genome, we need to do sequencing. This is one of the applications of sequencing. There are multiple applications, and you might be knowing that Human Genome Project was completed in 2003, and with the help of Human Genome Project, we came to know that there are around 3 billion base pairs present inside the human genome, and also that there are 30,000 genes that are involved in human genome, that are present in human genome, that code for different types of proteins and are responsible for doing different things in human beings. Okay, so this was the need of sequencing, and in case of Sanger sequencing, we use a very exciting or a very interesting method by which we can sequence, or we can know the exact position of different nucleotides present inside the genome of a person, of an individual, right? So let us consider the second point, dideoxy or chain termination. So these are the alternative names that have been given to this method, this Sanger sequencing method. Sanger sequencing method is also called as dideoxy method or chain termination method. We would understand this, or we would come to know about this in much more detail when we proceed in detail about the lecture. Why is it called as dideoxy method? I have written here dideoxy method. Now, why is it called as a dideoxy method? To understand this, we will have to go to the structure of the deoxyribose sugar. Okay, so we are talking about DNA, so we have to consider the deoxyribose sugar. You know that RNA consists of ribose sugar and DNA consists of deoxyribose sugar. So this is the first, and this is the second case, right? So in the first case, this figure that you are able to see here is the normal deoxyribose sugar. What is the difference between deoxyribose sugar and a ribose sugar? You would be knowing that if you number this carbons present in this structure, then this would be number 1, this would be number 2, this would be number 3, this would be number 4, and this would be number 5. Okay, so in case of the second carbon, you are seeing that an H is present over here, and H is present over here. Okay, so this H is the basic difference between the RNA and the DNA, that is deoxyribose sugar and ribose sugar. In case of deoxyribose sugar, at the 2' position, H is present, H and H is present, but in case of ribose sugar, here OH is present. Okay, so when OH is replaced by H, here you can see that OH, I am saying that here in case of ribose sugar, OH is present at this position, but in case of deoxyribose sugar, H is present. That is why the name deoxy is given, because oxygen is not present over here. Deoxy means oxygen is not present, therefore only hydrogen is present there. That is why it is called as deoxyribose sugar. So here it is called as deoxy, that you would have understood, I guess. In the second case, I have explained what is the meaning of dideoxy. Okay, what is the meaning of dideoxy? Here deoxy, you have got it, I guess. And dideoxy means that here also in the 3' position also, the OH which was previously present was now replaced by H. Let us understand this again. At 3' position, OH was present, and this OH has been replaced by H at 3' position over here. So this is 3', this is 2', this is 1', and this is 4, and this is 5. We are numbering the carbons, right? So at 3' position also OH has been replaced by H, therefore it is called as dideoxy. Di means 2. Two OHs have been replaced by 2 H's, therefore it is called as dideoxy. Okay, so after understanding the concept of dideoxy, let us understand that where it takes us. What changes would occur after making the alterations or after replacing the OHs with H. In case when you put dideoxy, when you make use of dideoxy instead of deoxy, what changes would take place? So you might be knowing that for joining two sugars together, you need a phosphodiester bond. Okay, so when you join, suppose let us understand this, when you join, this is one sugar and this is another sugar, you need to join this 3' end of this sugar. Suppose this is the 3' end of this sugar and this is the 5'. This is the 5' end of another sugar. Between these two, there is a phosphodiester bond that is present. Okay, in order to make this phosphodiester bond, 3' OH is needed. Okay, let us understand this, that in order to make the phosphodiester bond, what is required? 3' OH is required. Free 3' OH is required. But when you make use of this dideoxy, what are you getting? At 3' end, you do not have a OH. You do not have a OH. You have a H which has replaced that OH. So no 3' OH, no 3' free OH end would result in no extension of the sugar, no formation of the phosphodiester bond, and no another sugar would not be able to come and attach with the previous sugar molecule with the help of a phosphodiester bond. Let us understand this in much more detail in the subsequent figure. Let us understand this. This is the first sugar molecule. Okay, so that is what I was saying, that here free OH group should be present. So here O was present, and now the H that was present here was replaced by this phosphate group, and this links the second sugar to the first sugar with the help of this. This is the phosphodiester bond. So this is the phosphodiester bond. It is linking the first sugar with the second sugar. Here the 3' at the 3' end, OH was present, which was used to form the phosphodiester bond. Okay, but now if you understand this, then you would again come to know that when this kind of a group is present, when the dideoxy group would be present, then this free 3' OH group would not be present over here, and therefore phosphodiester bond would not be able to form, and chain extension or the linking of another sugar chain extension would stop. Okay, so this is the principle of Sanger sequencing. We would understand this in much more detail when we proceed into the lecture. So let us now go, after understanding the principle, let us now move forward towards the procedure. Okay, now let us understand the procedure. Suppose this is the sequence, or this is the DNA that you want to, this is the DNA strand that you want to sequence. Okay, so DNA is in double stranded form. you need to denature that DNA. What is the meaning of denaturation? Denaturation means separation of two DNA, separation of the two strands of the DNA. So here separation has taken place and single stranded DNA has been obtained, right? How can you denature a DNA? You can do that by heating. So if I heat, then the strands would be denatured. Okay, so now I have got two strands. Okay, suppose I need to sequence this particular strand. I am in need of knowing the position of each nucleotide in this particular strand. Now what would I do? I would make 4 tubes. Okay, each tube would be responsible for giving me the information about a particular nucleotide. Let us label these 4 tubes with different nucleotide names. Suppose this is the first tube. This would give us the information about guanine. This is the second tube, would give us the information about adenine. Third would give us the information about thymine, and fourth would give us the information about cytosine. Okay, what do we mean by give us the information? It will tell us that what is the position of cytosine in a particular DNA, in this particular DNA strand, right? In this tube, in each of these tube, what all components would we add? Let us understand this. In the first case, we would add all the DNTPs. What do we mean by DNTPs? DNTPs means A, T, G and C. We would add all the DNTPs in each tube. This means A, T, G and C would be added. A, T, G and C would be added in each particular tube. Next, what do? We add radio labeled DNTP. With DNTP we mean here that whatever nucleotide you want to know about, you would radio label that particular nucleotide. So suppose in the first tube I am aiming at knowing about the position of guanine. So I would label the I would label the DGTP only. Similarly, if I want to know about adenine, in the second tube I would radio label DATP. Next I would label DTTP, and in the next I would radio label DCTP. I would add all the DNTPs. That is fine. Please do not get confused about this. I would add all the DNTPs, but I would radio label that particular DNTP which I want to know about. Next, I would add DNA polymerase and I would add the template. Furthermore, finally, what is the most important thing that should be added over here is DDGTP, that is dideoxy, that particular nucleotide that you want to know about, right? Firstly, let us understand this again. I don't want that you guys get confused. All DNTPs are added. One thing, this is the first thing, radio labeled, only the radio labeled DGTP would be added. That means that particular nucleotide that you want to know about should be radio labeled. Next, DDGTP, the nucleotide that you want to know about should also have dideoxy version of the nucleotide, but that should be in very less quantity as compared to the normal DNTPs. Okay, that should be less in amount. Next, DNA polymerase should be added, and next the template should be added. So this would be same in all tubes. I would like to tell you. One more thing I would like to explain to you here, that this radio labeled GTP, an alternate of this can be the radio labeled primer. Suppose what I have to use a primer here because DNA polymerase cannot start to make the complementary strand of the template that we want to sequence. Therefore, we have to use a primer. What is a primer? Primer is a short segment of an RNA that would be used to extend the DNA fragment, right? So either you use the radio labeled GTP over here, or you used a radio labeled primer over here. What is the use of radio labeling over here? How would you know that this is my particular sequence? You need to locate your particular sequence, right? That is why you need to radio label each sequence. You need to understand that where is my sequence. You need to know that where is my sequence. That is why radio labeling is essential. Okay, so let us now understand this. We have provided all the conditions in order to function, in order to make the DNA polymerase make a complementary strand to this particular strand. This is the template strand that we want to analyze. We have given all the conditions to the DNA polymerase. We have given the DNA polymerase, we have given the template strand. This is the template strand. We have given the DNA polymerase, we have given all the 4 DNTPs, and we have also given the DDGTP. We have also given the dideoxy version, although the dideoxy version is lesser in quantity as compared to the normal DNTPs. Fine. I guess this would have been clear to you. Now, after giving this, what will happen? Let us take an example, okay? Suppose this is our chain. Suppose this is our chain A, T, T, G, C, G. So this was our fragment, this was our DNA strand that we wanted to sequence, okay? So in the first case, what will happen is, in the first tube, what you will get is, you will get the complementary strand to this, and you will get the complementary strand here, and here either you would get a G, that is the normal G, or you may get the dideoxy version of the G. Suppose you get the DDGTP. Suppose you get the dideoxy G over here. What will happen? The sequence would stop here itself, because no other nucleotide, no other sugar would come and join over here. Okay, because the phosphodiester linkage would be blocked, because no 3' OH free end would be present over here. Okay. Let us consider another scenario. This is another G. Suppose this you would get this fragment also in this tube. In the first tube, you would get this fragment also, this fragment also, and this fragment also. You would get all of the 3 fragments. Why? Because there is a chance that dideoxy version of this guanine can join over here and stop the chain, can join over here and stop the chain, and can join over here and stop the chain. So you do not know that at which position DDG would bind, or at which position normal guanine would bind. Okay, so you do not know about that. So you would get all the different fragments that have guanine and that have ending in guanine. Okay, so suppose you have got all these 3 fragments of varying length. Okay, I guess you would have understood this. Similarly for A tube, similarly for T tube, and similarly for C tube, you would get different fragments of different length, ending in here G, ending in here A, ending in here T, and ending in here C. Okay, now what do? You just take the entire contents of these tube and run a gel. You run a agarose gel. Okay, so suppose suppose this is the tube A, and you run the components of tube A in this lane, you run the components of tube T in this lane, tubes G in this lane, and tube C in this lane. Okay, so you would get different bands. You would get different bands. Why would you get different bands? Because you have different fragments. You have different fragments. In G tube only, you have suppose 3 fragments. Why? Because these fragments have different lengths. So you would get different bands on a agarose gel. Okay, you just have to analyze this gel and you would get your sequence. Now how would you get your sequence? Let us understand this. Suppose this was the agarose gel, and after analysis, when you use the what you have used radio labeled DGTP. So when you use to analyze this radio labeled GTP in this gel, you would get different bands. Okay, now how would you analyze these bands? Let us understand this. You might be knowing, you will be knowing that in agarose gel or in the gel electrophoresis, the fragment that has migrated to the lowest possible extent or migrated the highest distance, suppose this is the band that has migrated to the highest distance, it has come down to it has covered a lot of distance and come down to a lot of come down here. So, and it is I'm talking about this band, it has come down to the highest level and it has covered the highest distance. So we can say that this fragment or this band is smallest or is the least in compare when you talk about the length. So this is the smallest in length. Why I am saying this? Because in gel electrophoresis the migration depends upon the molecular weight or what is the amount or what is the length of that particular DNA segment. So the smallest or the highest the band that has migrated highest or that has covered the highest distance and that has migrated down would be the smallest. Okay, how can you understand this? Because how can you understand this? Because this is the primer. Okay, so the smallest band that is present, well, how will you call that smallest? That would be most near to the primer. Okay, the band that is smallest in length, that means that it would be most near to the primer. Here the primer was here, suppose this is the length, so this is the smallest. Suppose the fragment is of this length, right? So we can make out that this would be the smallest length of the fragment. Now similarly this and different kinds of bands are present in this gel. Now we just need to analyze. Okay, so what do we have to do? We have to put a 5' end over here at the lowest end. We have to take the 5' end. At the uppermost position we have to take the 3' end, and we just have to start labeling it. At 5' end, the starting was with adenine, so I have put an adenine over here. Next it was thymine, I have put a thymine over here. Just read the nucleotide in front of just read the nucleotide, respective nucleotide corresponding to the band that is present over here. So this was adenine, this was adenine, this was thymine. Again the next one is thymine. Again the next one is thymine, that is I have again put it over here. The next one is guanine, I have put it over here. The next one is adenine, I have put it over here. The next one is just after this is cytosine, this is like here. Next one is guanine here and adenine is here. So this is, and then after that we put the 3' end. We have started at 5' end and we have completed and we have reached at 3' end. Starting from 5' end, we just put the different names of the nucleotides that are present: A, T, T, G, and then again A, C, G and A. So this was the sequence. But remember that this sequence is of the complementary strand. We had this template, right? This was the template. This was the template for us, and we have just known the complementary strand. This is the complementary strand for this particular template. We know the sequence of this particular strand, and we aimed at knowing the sequence of this strand. So how would you know the sequence of this strand? We would just prepare, or we would just make the complementary strand to this particular strand. How would you do that? This was 5' end, so the complementary strand to this strand would be 3, starting from 3' end. Adenine, complementary to adenine would be thymine. Similarly, adenine, cytosine, thymine, guanine, and then finally you would put a 5' end. Let us understand this. The strand complementary to this strand. Why are we saying complementary? Because the sequence that we have obtained is of the complementary strand, not of the template strand. How would we get the sequence of the template strand? We would get the sequence of template strand by making the complementary sequence to this. How can we make that? Starting from just 5' end would be converted into 3' end, and then you would make the complementary strand adenine, thymine, thymine, adenine, thymine, adenine, guanine, cytosine, adenine, thymine, cytosine, guanine, guanine, cytosine, adenine, thymine, and then you would make the 5 and you would write the 5' end. So this is the sequence of the strand that we started with, or that we aimed at sequencing. So this was all about Sanger sequencing, and I hope that this would be useful to you. If you like my lecture, then please click the like button and subscribe to my channel. If you have any queries related to this particular video, please comment on the comment section and I would resolve it in the upcoming lectures. Thank you so much. Have a great day.